Assalamu alaikum students. In the last lecture we were talking about the process of diffusion across the cell membrane. Diffusion in as a word, it is the spread of any solute when it is placed in a solvent, right? When a pinch of salt is added into water molecules, then it is scattered throughout the water molecules. So the process of simple diffusion. Similarly, when you spray a small a scent in any corner of a room, the smell of the scent or the perfume, it spreads in the whole room through the process of diffusion. This is again what is shown in this picture. Drop of dye is placed in water and dye molecules diffuse into water, and water molecules, you can say, they diffuse into the dye molecules. Both the molecules are evenly dispersed in the last beaker. The movement of molecules in solution from a high concentration of that molecule in the direction of a low concentration of that molecule until equilibrium is reached is the process of diffusion. Diffusion across the cell membrane, it happens through this oxygen, carbon dioxide or extracellular fluids. They can be transported in and out from the cell through the process of diffusion when there is high concentration of any of the component. So here's a question: why does passive diffusion through the lipid layer only occurs with gases and small lipid soluble molecules? So कहना ये है कि क्यों गैसेस या फिर स्मॉल लिपिड मॉलिक्यूल्स ही ट्रांसपोर्ट क्यों होते हैं थ्रू सेल मेम्ब्रेन बाय द यूज़ ऑफ लिपिड लेयर ओनली. तो रीज़न क्या समझ में आ रही है? क्योंकि स्मॉल लिपिड मॉलिक्यूल्स ही अटैच हो सकते हैं लिपिड लेयर के साथ, और फाइनली लिपिड लेयर उन्हीं को एक्सेप्ट करके आगे ट्रांसफ़र कर सकती है. गैसेस का भी कुछ ऐसा ही कॉन्सेप्ट है, कोवेलेंट बॉन्ड्स के थ्रू लिंक्ड होती हैं, तो आयनिक चैनल्स नहीं होते हैं ये, तो ये भी लिपिड्स नॉन पोलर लेयर के साथ इंटरेक्ट कर सकती हैं. सो these substances, first option is these substances are non-polar, so is the lipid layer. These substances are polar, and so the lipid layer. No, these are non-polar. These substances are non-polar. Lipid layer is polar. No, lipid layer is non-polar. And then so the first option is correct. These substances are non-polar, and so the lipid layer. Facilitated diffusion, it is the process when diffusion is carried out, or the in and out or movement of the solutes or any of the component in excess, it is carried out through any of the other molecule than lipid layer. Lipid layer in the cell membrane. So this is the proteins, they form hydrophilic channel. So the proteins, any of the polar component like water or any ionic components like salts, they can diffuse through these proteins, and this is a process of facilitated diffusion. Why are channel proteins necessary to move hydrophilic ions through the membrane? Because they are polar and they have polar ends, and they can easily control the movement of polar compounds or ions or salts in and out of the cell. What are channel proteins necessary to move hydrophilic ions through the cell membrane? Ions are charged, they cannot go through the non-polar lipid layer. I think this seems to be correct. Let's see. Yes, ions are charged molecules, and ions are charged moieties, and that's why they cannot go through the non-polar layer. They are repelled by the lipid layer. Passive transport by facilitated diffusion. This is kind of the passive transport, how carrier protein it binds with the um, binding site it is having, and it binds with the specific molecule, and it can allow the transfer of this specific molecule from out of the cell to into the cell, and vice versa whenever required. And you see here amino acids, small sugars, proteins, they can enter through these carrier proteins, and in the third part, carrier protein changes shape, transporting molecule across the membrane, and in the fourth part, you see it retains, it resumes its original shape. Osmosis. Osmosis is the process of similar to the process of diffusion, but there is the insertion of a semi-permeable membrane. From here molecules can move to the either side. So the diffusion of water through a semi-permeable membrane is called osmosis. Water moves from its own high concentration to its own low concentration. Which direction will water move? In here you can see water will move to the concentration to the right side, where the concentration of the solute is higher. If the tonicity, if the contents of the cell and the fluid around the cell have the same concentration of dissolved solutes, this is called the tonicity of the cell. When the concentration in and out of the cell is same, whenever the fluid is isotonic compared with the cell, water moves in and out in equal amount. You see arrows are of the same strength. If the fluid around the cell, you see here, this is the cell, and around the cell is the fluid, it contains less dissolved solute. This is the solute, yellow ones. Then the fluid in the cell, so how water will move? According to the process of osmosis, water will move where its concentration is low. So it will move more into the cell, and the fluid is hypotonic. Fluid is hypotonic outside the cell, and more water enters cell than leaves. The cell may swell to the point of bursting here. And so here, if the fluid around the cell contains more dissolved solute than the fluid inside the cell, the fluid the fluid is carrying more solute outside the cell, so the more water leaves the cell than enters, and in this way cell will shrink. This is the whole process of tonicity represented in a single slide. What is the tonicity of the solution that these red blood cells have been dropped into? In the first left picture, equal amount of water moves in and out of the cell, so the red cells are at the equilibrium stage. In the second slide, the movement of water out of the cell is higher because of the hypertonic condition, and in the third slide, due to hypotonic, more solute is inside the cell, and that's why water is moving into the cell, and the cell is swelling. Isotonic, hypertonic, and the last one is hypotonic solution. Again, this is in a nutshell. You can see net water movement out, the cell is crenated. This is hypertonic condition. In isotonic, the cell is in the normal condition, and cell is swollen or lies when it is hypertonic condition, and arrow it shows the concentration of the solute in extracellular fluid, in the fluid outside the cell. So you can easily identify the cell, what will be the fate of the cell, either it will be shrinked or it will be lysed. So and this is the lecture for today, and it completes our lecture of cell membrane, and we will join in the next lecture, that is, I guess, about amino acids. Okay, see you.